Hi dear students, welcome to our discussion on the microbiology MCQs which were asked in the recently concluded INI CET exam. Looking at the MCQs, I felt it was a very nice set of questions in the microbiology part. If you have studied your notes very well, you know how to apply them, that application that you can get only by attempting MCQs in grand tests and subject-wise tests regularly. You can easily breeze through all the microbiology MCQs except for a couple of mistakes that were made by the examination committee itself, right? Apart from those two MCQs, it was a breeze, okay? So let's get going. A patient presented with a painless hard nodule in the neck. On examination, it was not warm to touch and it was easily mobile. Biopsy was taken and on anaerobic culture, molar tooth colonies grew in 2 to 3 days' time, which shows gram-positive branching rods. The first thing that should strike you whenever you read gram-positive branching rods, what are we going to think about? We are going to think about actinomycetes, the higher group of bacteria which are so characteristic in their morphology. Which of the following is the drug of choice? Now, when we think about actinomycetes, we're going to think about either actinomycosis, that is, which is caused by actinomyces. And what have we learned about them? That these are strict anaerobes. And the other group are the important one is nocardia. So what are we going to think looking at this strict anaerobe? That is what is mentioned, anaerobic culture. And gram-positive filamentous rods, that means this is definitely diagnosis becomes actinomycosis. So this is cervicofacial actinomycosis presenting as a hard nodule in the neck. Okay. So what is the treatment? The drug of choice is penicillin. Okay. So that is the drug of choice. Please do not confuse actinomycosis with actinomycetoma, which is caused by nocardia, which is a strictly aerobic bacterium. Okay, so these two important hints were given. Apart from that, actinomyces typically form molar tooth colonies. Okay, when you grow them on a blood agar medium or a nutrient agar medium, that's the type of colonies that they form, which look like molar teeth. Okay, that's the typical morphology, gram-positive filamentous rods showing branches. Okay, right. Let's move on to our next question. We got 3 questions on genital ulcers. Which amongst the following organism causes a painful ulcer on the glands? Chlamydia trachomatis, Haemophilus ducrei, herpes simplex, and Neisseria gonorrhoeae. The first and the last option are never causes of genital ulcers. They do cause urethritis, but not genital ulcers. What are the 5 causes of genital ulcers? 2 of them are painful. That is soft sore or Shankroid caused by Haemophilus ducrei, and the other is herpes genitalis caused by the herpes simplex virus 1 and 2. Right? So our answer out here, out painful ulcers on the glands, the answer is B and C. Coming to the other 3 causes of ulcers, which are painless. What are these? These are granuloma inguinale, syphilis, and lymphogranuloma venereum. Right? So I've made this table. So quickly let us brush through all the important causes of genital ulcers. So the first through are the painful ones. Genital herpes caused by HSV2 more than HSV1. Characterized by painful multiple groups of vesicles or ulcers associated with tender non-suppurative lymphadenopathy. Diagnosis is done by taking a scraping from the base of the ulcer or the vesicle, preparing a Zank smear on which you're going to see multinucleated giant cells. Next step, direct fluorescent antibody testing for antigen detection. Then we can culture them on cell lines, or we can grow them on a chick embryo on the chorioallantoic membrane. And finally, most sensitive would be the nucleic acid amplification test, that is the PCR, right? Next is Shankroid, which is caused by a gram-negative Coccobacillus Haemophilus ducrei. Here the Genital ulcer is a non-indurated, shallow, tender ulcer with ragged edges. That's why it is called a soft sore, and it bleeds very easily on touch. It is associated with tender suppurative lymphadenopathy, and when we do a microscopic diagnosis of, examination of the exudate, we are going to see these bacteria are arranged in parallel chains. That this is typically described as school of fish appearance or railroad track appearance. Next, we can culture it on chocolate agar supplemented with isovital X with fetal calf serum. And finally, most sensitive would be the nucleic acid amplification tests. Next, we move on to the 3 causes of painless genital ulcers. First is syphilis, which is caused by Treponema pallidum, subspecies pallidum. It's a non-tender, indurated ulcer with raised regular edges, associated with non-tender, rubbery, non-suppurative lymphadenopathy. Diagnosis is done by direct fluorescent microscopy of the exudate from the hard chancre, or antigen detection by direct fluorescent antibody testing, most sensitive nucleic acid amplification tests, but most commonly what is used for diagnosis are the serological tests, the non-treponemal and the treponemal tests. So the next one is lymphogranuloma venereum caused by Chlamydia trachomatis L1, L2 and L3. This is associated with a painless small genital ulcer which heals very rapidly. It is associated with tender suppurative lymphadenopathy. Many times these lymph nodes break onto the skin forming draining sinuses, and what we can sometimes see is the groove sign of Greenblatt. I will show you the picture soon. So groove sign is seen in LGV. Diagnosis on microscopy, the elementary bodies of the Chlamydia trachomatis are called as Miyagawa granulocorpuscles. Then we are going to see the most sensitive would be the gnats, and lastly we can do a serological diagnosis. Lastly, we come to granuloma inguinale caused by Klebsiella granulomatis, a gram-negative rod. So associated with painless papule nodule or an ulcer, associated with what are called as pseudobuboes because of the indurations produced in the subcutaneous tissues. These are not actually lymphadenopathies. On microscopy, we can see the Donovan bodies are the typical bipolar staining of Klebsiella granulomatis. This bipolar staining is also seen of Haemophilus ducrei when we stain them on gram stain, right? So that is about genital ulcers. Let's take up our next question. The following are true about Shankroid. We just learned, yes, we see the school of fish appearance. Can you see that? The school of fish in the exudate, that's the school of fish. How fish of the same breed, they move along in oceans to, evade their enemies. That's how they move around. So that is called a school of fish. It's a coordinated, organized movement. So it bleeds on touch. That's why Shankroid is also called as soft sore. It is painful. Yes, it is painful. So first, second and third are correct, but groove sign is seen in LGV. So our answer here is option B. A, B and C are correct. Let's move on to a picture of LGV as I promised. That's the groove sign of Greenblatt. That is the inguinal lymph nodes are enlarged. So we have the inguinal ligament passing, and above and below it is the other lymph nodes which are enlarged. So that is producing a groove on the lymph nodes. Let's move on to question number 4. A young male presents to the STD clinic with painful lesions as shown in the image. So we have again painful genital lesions. And can you see there are multiple lesions? So this is more likely pointing towards herpes genitalis. What is the most likely microscopic finding of the lesion? What are we going to do? We are going to scrape off the roof of this vesicle, take a scraping from the base and prepare the Zank smear. And what are we going to see on microscopy? Multinucleated giant cells. So our diagnosis here is herpes genitalis, and we are going to prepare the Zank smear. Okay. Okay. Henderson-Peterson bodies, these are seen in molluscum. But in molluscum, we do get these kinds of lesions, but these are not painful lesions, okay? And Halberstetter-Provazek bodies, these are seen in trachoma. And Punt cells, these are seen in granuloma inguinale. Okay, so these are cells which are having the Donovan bodies inside them. So due to Klebsiella granulomatis, right? But that is against because it is painful, right? So our answer here is option A. Question number 5. Affinity maturation is due to which of the following? The answer is option A, somatic hypermutation. Okay. Now, this is a slightly tough question, but if you've listened to my lecture on immunity carefully, I have talked about affinity maturation when I talked about the types of antigens, the T-dependent and the T-independent antigens. But let me explain it once more here. So what happens is whenever B cells are activated in response to a particular antigen, initially we know they produce IgM antibodies, and these are low affinity antibodies. What do we mean by affinity? The strength of the bond that forms between an antigen and the antibody. So these are initially low affinity, but on a prolonged exposure to the same antigen or on repeated exposure to the same antigen, I think that was what was mentioned in the option, repeated exposure to the same antigen, what happens is those activated B cells in the lymph nodes, in the germinal centers, they will form special areas which are called as the dark areas, dark centers. Here what happens is they will undergo this phenomenon, somatic hypermutation, in which what is happening is random point mutations are occurring in the B cells, so that these random point mutations are occurring, hoping that one of these point mutations will be so useful for that B cell that it is now able to bind to that antigen with higher affinity. Right? Now, these are random mutations. Some of these mutations may be such that they are totally useless. Now the B cell has no affinity for the antigen or still has low affinity for that antigen. So it's a totally useless mutation. But some mutation may be such that this B cell now can bind to the antigen with higher affinity, right? Now, what is going to happen now? Now there is a special cell in the lymph nodes, which is called as the follicular dendritic cell. Its job only is to select out only those B cells which are producing higher affinity antibodies. So as the B cells which have undergone somatic hypermutations are moving out of those dark zones, the follicular dendritic cells on their so many dendrites will present the antigen. The antigen will be presented to that B cell. Do you bind to this antigen with a higher affinity? If the B cell has no affinity or still has low affinity for that antigen, the follicular dendritic cell says, you are useless. I want a higher affinity one to be produced. So those B cells which are producing higher affinity, they are binding to that antigen on the dendrites of the follicular dendritic cell with higher affinity. The FDCs will give them a survival signal. You are the only ones who will survive this. Will now survive. So only those B cells will survive which are producing higher affinity antibodies, right? So this shift from low to high affinity antibodies is called as affinity has got matured, right? So this is a small and simple picture of how affinity maturation is occurring. That's our antigen. That's our B cell which has got activated. After the somatic hypermutations, which were the point mutations, random point mutations, now the follicular dendritic cell is going to select out only those B cells which are producing higher affinity antibodies, and that is the one which is surviving. Rest, all of the B cells which have undergone that mutation will be given a negative signal. They will undergo apoptosis. Okay, this is a beautiful picture of how affinity maturation is occurring. That's our activated B cell. It has entered this dark zone of the germinal center, undergone somatic hypermutations, and as it is moving out into the light zone, the follicular dendritic cell are presenting the antigen and selecting out only those B cells which are producing higher affinity antibodies. And now these activated B cells will go to another cell. This cell is the follicular helper T cell. This follicular helper T cell will now give it another signal in the form of binding of CD40 to CD40 ligand, and that is going to help this B cell to undergo class switching. That is, earlier it was producing IgM antibody, now it starts to secrete the other classes of antibodies. So what is happening? Class is getting changed, hence it is called as class is getting switched, or isotype switching is occurring. I will talk about it also just now, right? Have we understood the affinity maturation? It is because of somatic hypermutation. And what's the important cell which is going to play an important role in affinity maturation is the follicular dendritic cell. So FDCs are exceptions. They are not antigen presenting cells, but they are responsible for affinity maturation. Let's read the other options in the question. I'll talk about isotype switching later. Let's talk about VDJ rearrangement, because many students, I think, have answered this. VDJ rearrangement is responsible for the antibody diversity, the huge diversity of the antibodies produced by different B cells. That is because of the VDJ rearrangement. If you look at this, that's our, of course, you all know, I'm sure, that B cells, what receptor do they have? The antibody that they are going to secrete later is already present on their surface as the B cell receptor. And this is of two classes, IgM and IgD. Right? That's in a mature B cell. So when a B cell is undergoing maturation in the bone marrow, what happens is for generating that antibody which is going to act as a B cell receptor, VDJ rearrangements will occur for the heavy chain synthesis, and VJ rearrangements will occur for the light chain synthesis. So this is our VDJ rearrangement. Look at it. It looks pretty complex, but it is not. It's pretty simple if you try and understand it. So this is our chromosome number 14, which is encoding the heavy chain. So we have three special gene loci, which is going to encode for the variable part of the heavy chain. So V contains as many as 65 genes. The D gene segment contains as many as 27 genes, and J gene segment contains as many as 6 genes. So what is rearrangement is a special group of enzymes will come, RAG1 and RAG2 will randomly select out one of the segments from the J, one of the genes from the J and the D, and join them together. It is totally random, right? Once this has rearranged, one of these 65 genes will be selected out randomly, and finally we will have a complete gene which will be encoding for the variable part of the heavy chain, right? So this is called as VDJ rearrangement. So can you think of the possibilities? 65 multiplied by 27 multiplied by 6 is the number of possibilities of the variable region. During the joining of these genes, what happens is randomly some nucleotides are deleted, randomly some nucleotides are added, and that is going to create further diversity. So at a particular point in time, these are the number of possibilities. This was just 10 raised to power 4, but further diversity is occurring because of those random additions, p nucleotide addition, etc. Those are called as junctional flexibility, right? So that is VDJ rearrangements. Now, further ahead, we will have the constant gene locus C mu, C delta, C gamma 3, and so on to encode for the constant part of the heavy chain. So if C mu joins up, what is going to happen? This is going to be the mu heavy chain. If the C delta is joined up with this rearranged gene, what are we going to get the product as? Delta heavy chain. If C gamma 3 is joined up, we're going to get gamma 3 heavy chain. Okay, so that is our heavy chain rearrangements. Once the B cell have rearranged their heavy chain, they are expressing the heavy chain on their surface as the B cell receptor. Now the RAG1 and RAG2 enzymes will go and rearrange the light chain genes. And here we have only 2 gene segments to encode for the variable part of the light chain. So we will have about 40 genes in the variable. In the joining gene segments, we will have 5 genes. Randomly, one of these genes will be selected out, joined up with each other, and then joined up with the constant region constant region gene, and we will have the complete gene for the light chain, right? So this is called as VJ gene rearrangements, right? So that is our receptor expressed. Complete receptor gets expressed. So these are all random phenomenon, and this is called as rearrangements, somatic rearrangements. Don't confuse somatic hypermutation with somatic rearrangement or gene recombinations. They are totally different things, right? So this is how complex we get the ability to secrete the huge diversity of the antibodies. Moving on to isotype switching. I hope you all know what are isotypes of antibodies. What are these? IgG, IgD, IgE. These are isotypes. How is each isotype of antibody different from each other? By the heavy chain. Isn't that so? If there is IgG, gamma heavy chain, it would tell us this is IgG antibody. If there is delta heavy chain, this would tell us this is IgD antibody. If it is epsilon heavy chain, it would tell us this is IgE antibody. So that is isotypes of antibodies. Now, how do B cells get the ability to secrete different classes of antibodies? Initially IgM, later IgG, IgA, and IgE antibody. That is called as isotype switching. And this occurs only when the B cells have undergone somatic hypermutation, as I showed you in the lymph node earlier, that a follicular helper T cell will come and give it a special signal. The CD40 of the B cell will bind to the CD40 ligand of the T helper cell, and that will tell it the B cell to undergo isotype switching. So earlier it was secreting IgM, now switches its class to secrete IgG and so on antibodies, right? So remember, this isotype switching is because of what is called as switch recombination. Class switch recombination. And which enzyme is responsible for this? It is activation-induced cytidine deaminase. AID in short. So any defect in this enzyme, mutation in the gene for this enzyme, means the B cell is going to only secrete IgM antibodies. Ever heard of hyper IgM syndrome? That is because probably one of the causes could be a gene mutation in this enzyme. Another important signal, as I mentioned earlier, is CD40 of the B cell has to bind to the CD40 ligand on the follicular helper T cell. So all this has to bind. So any defect in either of these expression again is going to lead to hyper IgM syndrome. Okay. So this is very important, class switch recombination. Right. So this is what is happening. Once the B cell have rearranged the receptor gene, they will always secrete IgM antibodies. So that means in the VDJ gene, what is going to always first get joined up with the rearranged gene, it is always going to be C mu, right? So that is why it is secreting IgM antibody. Isn't that so? But because of those signals, if the C mu, instead of C mu, what happens is it is C gamma 3 getting joined. Or it could be C alpha 1 joining up. So what is happening? It is IgG3 or it is IgA1 getting secreted by the same B cell. The variable part is remaining the same. The class is getting switched, right? So this is isotype switching. So those are the multiple genes which are present on the heavy chain gene locus, C mu, C delta, gamma 3 and so on. Either of these genes getting switched with the same variable gene. Okay, let's take up our next question. One of my favorite topics in bacteriology, teaching about diarrheagenic E. coli and their pathogenesis. So this is such a simple question if you've taken the lecture seriously. So ETEC, enterotoxigenic E. coli, they produce a heat-stable toxin. Toxigenic, labile toxin and stable toxin. So that is the answer. Fortunately, it has been arranged in the same way. Enteroinvasive E. coli, they invade the intestinal mucosal cells. Enterohemorrhagic E. coli, they produce a toxin which is acting on vascular endothelial cell, the verocytotoxin. That is, its site of action is this. And enteropathogenic E. coli, they adhere to the mucosal surfaces and cause attachment effacement effect. So our answer here is option first, right? So here is a beautiful diagram, and I'm just going to quickly revise all these important pathogenesis of E. coli causing diarrheas. Let's start with ETEC. It produces, first of all, what is it adhering by? What are those structures on its surface? The first virulence factor, colonization factor, antigens. And once it has adhered to the small intestinal mucosal cells, it is expressing either one or both of these toxins, heat labile, heat stable toxin. Heat labile toxin increases cyclic AMP, whereas heat stable toxin increases cyclic GMP. This causes the secretion of ions into the intestinal lumen. Next, moving on to EPEC, most common causes of bacterial infantile diarrheas. They have something which is called as the bundle forming pilus, and then they adhere to cup-like pedestals and cause effacement of villi. And they also disrupt the tight junctions between the enterocyte, increasing the permeability. Moving on to enterohemorrhagic or Shiga toxigenic Escherichia coli. There are two important virulence factors. One, they produce the same effect, attachment effacement effect, and then they produce the Shiga-like toxin, which acts on endothelial cells, inhibits the protein synthesis by breaking the 60S ribosome and thereby leading to necrosis of the endothelial cells. And when this Shiga toxin disseminates via the blood, it causes what is called as hemolytic uremic syndrome. And very important serotype of enterohemorrhagic E. coli is E. coli O157H7, right? Enteroaggregative Escherichia coli, common causes of persistent diarrhea beyond 15 days, and they cause what is called as, they first adhere by aggregative adherens fimbriae, and then they secrete a toxin which is called as enteroaggregative stable toxin, which also increases cyclic GMP, leading to the secretion of ions into the intestinal lumen. So this is a cause of persistent diarrhea. It is also a cause of traveler's diarrhea. Moving on to enteroinvasive E. coli, these are invading. So they cause disease similar to shigellosis. So first of all, they will they are sampled by the microfold cells, and then they attach to the basolateral aspect of the mucosal cells, and then keep on entering by secreting special proteins which are called as EPA proteins, invasive plasmid antigens or virulence marker antigens. Lastly, we have the recently characterized diffusely aggregative Escherichia coli. As you can see, their adherence is here and there. They mainly cause pediatric diarrheas. Okay, next question, diarrhea in Vibrio cholerae is due to disruption of which of the following? This is a repeat question. It was asked about 5 years back in AIMS exam. It is asking us about the zonular occludens toxin of Vibrio cholerae, right? So Vibrio cholerae, basically the O1 and O139, which cause the disease cholera, produce, of course, the cholera toxin, also called as the choleragen. They also secrete the zonular occludens toxin. The name itself is suggesting it disrupts the tight junctions between the enterocytes and thereby increasing the permeability. And lastly, accessory cholera enterotoxin. This also contributes to the secretion of ions into the intestinal lumen, right? Of course, the major virulence factor remains the phage-mediated cholera toxin. Here's a diagram just for us to revise the mechanism of action of cholera toxin. It's a AB5 subunit toxin which binds to the GM1 ganglioside receptors, undergoes receptor-mediated endocytosis. In the endoplasmic reticulum, the A subunit separates out, further gets split into A1 plus A2, and the actual active part of the toxin is A1, which now what it does, it causes the ADP ribosylation of the alpha subunit of the G stimulatory protein, which leads to increase in activity of adenyl cyclase, thereby increasing cyclic AMP, which leads to the continued phosphorylation of the cystic fibrosis transmembrane regulator. So all those ionic changes, secretion of chloride ions into the lumen and reduced absorption of sodium and chloride from the tip cells. Right. Question 8, our favorite, has become beta-1,3-D-glucan and galactomannan antigens in AIMS exams. Galactomannan antigen in serum is detectable in which of the following? The answer to this is only invasive aspergillosis. Here, let me just clarify, this is not beta-1,3-D-glucan antigen we are being asked about. This is an antigen which is present in the cell wall of all fungi, except mucormycosis, mucorales. So that means in systemic mucormycosis, this will not be detectable in the patient's serum. In all other systemic fungal infection, this antigen is detectable in the patient's serum. And one more exception is cryptococcosis. Again, beta-1,3-D-glucan is not detectable in cryptococcosis. Reason is because the capsule is enclosing the beta-1,3-D-glucan in the cell wall. Hence, this antigen is not released into the blood. In all other systemic fungal infection, beta-1,3-D-glucan is detectable in the patient's serum. But here the question is galactomannan, and this antigen is specific to Aspergillus. So it is only detectable in invasive aspergillosis. So our answer is A only. A patient presents with meningitis with a history of fever for 4 days with erythematous rashes on the chest. Meningitis of 4 days with rashes on the chest. There was no history of travel or tick bite. His CSF findings show increased protein, normal glucose. So this is going against a bacterial meningitis. Mild lymphocytosis, again ruling out bacterial meningitis, and few RBCs. Which of the following is the most likely organism? So of course these two are immediately ruled out. Options are West Nile virus and enterovirus. For your knowledge, West Nile encephalitis is reported in India. So yes, this could be a cause. But what is our answer? It is the most common cause of aseptic meningitis. Of course, our answer is going to be enterovirus. And especially some Ecovirus 9. Ecovirus 9, this is especially associated with a rash, right? So that is what is mentioned, and likely answer is this one, right? So I don't know why people you started to think of West Nile virus as the cause of this meningitis. Yes, it can cause, but here we are going to think of the 85 to 90% cause of aseptic meningitis, enteroviruses, right? So that is our answer. So just quick, this is a list of the important arboviral diseases which are reported in India. Japanese encephalitis and West Nile, their vectors are Culex. Dengue and chikungunya, these are transmitted by Aedes. KFD, Crimean Congo, Ganjam and Bhanja, these are transmitted by heart ticks. These are all reported from India. Sandfly fever and Chandipura encephalitis, of course, these are transmitted by sandfly. So this is an important list. Please remember, yes, West Nile, Crimean Congo, all these are important causes of our arboviral diseases in India. Right? So when we talk about aseptic meningitis, as in this case, of course, the most common cause remains enteroviruses, especially the Coxsackie B, ecoviruses, right? So these are the most common, followed by herpes viruses, especially HSV1 or HSP2, sometimes CMV, sometimes Epstein-Barr virus. Then we can have mumps meningitis. Then, of course, arboviruses like Japanese encephalitis, West Nile fever virus, and rarely Chandipura encephalitis. Right. Question 10, pneumonia due to a gram-negative pleomorphic bacillus. Okay, the morphology in a question is very, very important. Like we had that first question, gram-positive filamentous rod showing branching, only think of actinomycetes. Here, we are going to think of gram-negative pleomorphic bacillus causing pneumonias. What am I going to think of? I can think of Haemophilus. Growing on chocolate agar. Whenever chocolate agar is mentioned in the question, think of Haemophilus or think of Neisseria, right? And this is catalase and oxidase positive, right? Neisseria, we're not going to think of pneumonia first. We're going to think of Haemophilus as the cause, okay? So gram-negative pleomorphic bacillus growing on chocolate agar, catalase and oxidase positive. This is definitely not Pseudomonas as the cause because that is a gram-negative bacillus. Of course, it is catalase and oxidase positive. This chocolate agar word would not be mentioned in that case, in case of Pseudomonas. Actinomyces is ruled out because of its morphology. E. coli also again ruled out. It would be mentioned as gram-negative bacillus. So our answer is Haemophilus influenzae. So this is the reason it is very important to remember correlating things, morphology with the culture medium, with the biochemicals, then you can easily arrive at the answer. Now this was a sad question. So every time AIMS people are giving such a question which is going to fox everyone. So no need to get sad about it. I could not attempt. Nobody else would have attempted this question. Last time we got a crazy question, a graphic representation of leptospirosis. I'm sure no one got it. Similarly, I'm sure this one also no one. It has been picked up from an article on Zika virus neuropathogenesis. Okay. So here what was shown was something related to skin. You could see keratinocyte. It was such a small picture, you could hardly make it out. Skins, and you could see its transplacental transfer. So you have to think of transplacental, and it was marked as something affecting the brain. So you would think of a cause of microcephaly. If you read the answers, all of them are viruses. Nipah virus is not a cause of microcephaly. So the confusion here is between all these are causes of microcephaly. Apart from another causes toxoplasmosis, other causes varicella. These are the 5 important causes of microcephaly: toxo, varicella, Zika, rubella, and CMV. Now, looking at this picture, if it is entering through the skin, that means it is being inoculated through something biting. Out of these biting, the only one which is an arbovirus, which is being inoculated by the bite of the Aedes mosquito, is Zika virus. Rest all are never acquired through the skin. Toxo, varicella, rubella, CMV, neither of them. This is a sad question, but you got to. So this is Zika virus. Try to read this. It was way above, everything in this article went way above. Some cytokines, special cells which are called as Hofbauer cells. These are the macrophages in the placenta. So Zika virus, that's the transmission cycle. So in the sylvatic, the jungle cycle, it is the primates, the monkeys, which are acting as the reservoirs. In the urban cycle, it is just transmitted from human to human via the vector Aedes aegypti and Aedes albopictus, right? And Zika virus came to worldwide attention way back in 2015 and 16, when it was reported to cause several hundreds of cases of microcephaly. So it was not given much attention. It was just known as a cause of fever with joint pains, but simultaneously several cases of microcephaly from females who had been reported to develop Zika virus infection several months ago were reported at that time. So that is the congenital Zika syndrome. So mostly it has been found, though a few cases have been reported also in the later part of pregnancy, but predominantly the transplacental transmission of Zika occurs in the first half of pregnancy. So microcephaly with a partially collapsed skull, thin cerebral cortices with subcortical calcifications, microphthalmos, macular scarring. So something is very typical about Zika: the subcortical calcifications, the macular scarring, the congenital contractures, and the marked early hypertonia. These are all components of the congenital Zika syndrome. The correct order of steps in PCR reaction are which of the following? If you don't know this, it is very, very sad. So the steps are denaturation, followed by annealing, followed by extension. Okay, so the template DNA, double-stranded DNA, first it is denatured. The two strands are separated from each other by raising the temperature to somewhere around 94 to 95 degrees Celsius. What is PCR? Basically, we are amplifying a target sequence which is called as the template DNA. After the step of denaturation, we have already added primers. What are these? These are the forward primers and the reverse primers. Two primers are added, which are complementary to the bases, the nucleotides which are present in the initial part of the template DNA. So on one strand, and the reverse primer is complementary to the nucleotides at the end of the template DNA on the opposite strand. So when the primers bind, so once the temperature has been raised for a specified amount of time, denaturation occurs. Next, we are going to lower the temperature to 50 to 55 degrees Celsius. That will lead to the binding of the primers. That is called as annealing. Now we're going to raise the temperature to 72 degrees Celsius, because the optimum temperature of action of the enzyme responsible for the amplification is the Taq polymerase. What is the source of the Taq polymerase? Thermus aquaticus. Why are we using Taq polymerase in the PCR cycle? Because this is the polymerase enzyme which can survive that high temperature we are initially raising the buffer solution to during the step of denaturation. It can still survive. It does not get denatured at this high temperature. That's the reason we are using Taq polymerase in PCR. So then at this temperature, it is going to extend these primers by keeping on adding the nucleotides which have already been added in the buffer solution, right? So those are the steps of PCR. Which protein is present in the HPV vaccine? Right, so many students made the blunder of marking this as the answer. These are the oncoproteins of papillomaviruses. E6 inactivates the P53. E7 inactivates the protein, that is the retinoblastoma protein. So these are the tumor suppressor proteins they are inactivating. Obviously, we're not going to give them in the vaccine. What are we giving? The capsid protein. And what's the major capsid protein? The L1. This is the recombinant L1 protein, which we are giving in the Cervarix and the Gardasil vaccine. Recombinant L1 protein. So in Cervarix, the recombinant L1 protein has been synthesized in insect cell lines. And I'm sure you all know what it contains, the L1 protein of 16 and 18. It is just a bivalent vaccine. And the Gardasil, here the recombinant L1 protein has been synthesized in the baker's yeast, that is Saccharomyces cerevisiae. And today, at present, which one do we have? Gardasil 9. The 9-valent vaccine, which is providing protection against 6, 11, 16, 18, 31, 33, 45, 52, and 58 genotypes of human papilloma viruses, right? So that's our human papilloma virus genome, which is encoding 2 structural proteins, L1 and L2. L2 is the major protein. The minor capsid protein is this one, right? L1 is the major one. And those are our oncoproteins. There are 6 non-structural proteins, out of which E6 and E7 are the oncoproteins. We've already discussed about them, right? Which of the following diseases has the highest mortality once clinically manifested? So easy. Rabies. Once the patient develops the disease, it has 100% mortality. Ebola as high as 90%. Which is the Ebola species which is associated with maximum mortality? The Zaire one. Zaire Ebola virus. Nipah virus, yes, has 40 to 70% mortality. It was as high as 90% when the outbreak occurred in Kerala in 2018, but that's the average mortality of Nipah virus. Zika has hardly any mortality associated, just a mild febrile illness. So our answer here is rabies, 100% fatal. A 30-year-old patient presents with urethral discharge. The gram stain picture is given below. What could be the probable diagnosis? Now, this is an image which is quite a favorite of AIMS faculties who set the paper. What can you see? Intracellular gram-negative diplococci, right? So this is going to be Neisseria gonorrhoeae, right? So that's our answer, gram-negative diplococci. Okay. Trichomonas vaginalis, you would see the trophozoite. Gardnerella vaginalis, those are seen as gram-variable rods. Chlamydia trachomatis cannot be demonstrated by gram stain. You have to stain it by Giemsa, the inclusion bodies. So this is, of course, gonococcus as the cause of the urethritis in this 30-year-old male. The same figure is also shown for meningitis, then think of meningococcus. Transmission cycle of which of the following is shown in the figure? Now, carefully, if you look at it, you can easily catch. In the center, you are seeing something which is spiral. Then you come to the rodents. Something is being passed out in the rodent excreta or in the urine. Of course, in lepto, it is urine. Then contaminated mud and soil while you wade through water, which is like flooded water, or while water sports, fishing, bathing in contaminated water. That's how it enters the skin, okay? Through breaks in the skin or swimming, exposure of conjunctiva or mucous membranes. Then we have so many animal reservoirs, dogs, horses, cows, sheep as acting as reservoirs, right? So this makes them the people who are responsible for slaughtering animals, these are people also at risk. So answer here is leptospirosis. So some of you had asked me, why not hantavirus? Hantavirus is, yes, the reservoirs are rodents. But what are you seeing right in the middle? You can see this leptospira morphology in relation to rodents transmitted by rat urine. So, of course, hanta is easily eliminated. By the way, hantaviruses are acquired by inhalation of rodent feces or rodent urine or the bite of rodents, not through the skin. They would have otherwise made an arrow like that, and you would not have so many domestic animals acting as the reservoirs. So that's our leptospira, the typical morphology, the closely wound spiral with either one end hooked or both the ends hooked. What is this? This is the Dinger's ring. When it grows in EMJH, Korthoff's, Fletcher's or Stewart's medium, it forms a ring of growth just beneath the surface of the medium. So leptospira, unlike Treponema pallidum, is very well cultivable on cell-free media. Identify the parasites shown in the figure. So these are huge parasites that you can see naked, and it's a thick fleshy worm. Such huge worms are definitely Ascaris. Let's read the options. Ankylostoma duodenale, the hookworms. These are very tiny, about 1 centimeter in size. Enterobius vermicularis, again 1 centimeter, sometimes even 3 millimeters. So these are also too tiny. It's not the answer. Strongyloid is the smallest roundworm found in the human small intestine, 1 to 3 millimeters in size. So answer is Ascaris. What are these worms? Are these males or these are females? The posterior ends are curved. Both these are male Ascaris lumbricoides. So these are pictures of Enterobius. I don't think you can be asked. These are very tiny. This is looking like a thread. This is threadworm Enterobius. These are the hookworms, very, very small, 3 millimeter to 11 millimeters in size. These are the hookworms. Just compare the size with a matchstick. These are so tiny. So these are, can you identify this? This is an interesting picture. What is this? Looks like a whip. This is Trichuris trichura. The posterior end is broad. The anterior end is very slim. A farmer presents with a swollen foot and multiple sinuses. Okay, this is a farmer with multiple sinuses. The moment you read sinuses in a person who is at risk of that acquiring the traumatic implantation of a fungus, this is definitely Mycetoma. In sporotrichosis, what is characteristically mentioned in the questions is nodulo-ulcerative lesions, not sinuses, right? So this is definitely Mycetoma. Mycetoma, as we all know, is of two types depending upon the cause. Actino is caused by aerobic actinomycetes, which are Nocardia, Streptomyces, and Actinomadura. Here, what the granules that you're going to see in the sinuses that are formed are either off-white or they are yellow colored, except in one, Actinomadura pelletieri, in which they are red in color. And the treatment in actinomycetoma is the Welch regime, that is the amikacin given with cotrimoxazole. The fungal cause of mycetoma are pheoid or hyaline fungi, pheoid meaning pigmented, the dimatiaceous fungi, Madurella and Exophala, the hyaline fungi, Pseudallicheria, Acrimonium, Aspergillus. Okay. Depending upon the cause, in case it is a pigmented fungus, you're going to see brown or black granules. If it is a hyaline fungus, it is going to be off-white or yellow granules. The treatment is azoles with debulking surgery. In the world, actinomycetoma is much more common. The ratio is 60 versus 40 of eumycetoma. In India, the ratio is 65-35. Most common cause of mycetoma in India is Actinomadura madure. Match the following with their organs of locomotion. Right. So this is why it is important to know why I talk when I teaching you guys, I tell you, okay, this is phylum Sarcomastigophora, subphylum Sarcodina, subphylum Mastigophora. This is containing the pseudopods, this is containing the flagellates. The reason is this, such a simple question. Unfortunate if you don't attempt such a simple question on morphology. Balamuthia mandrillaris, this is an amoeba. So this is going to be motile by pseudopodia. Balantidium coli is a celiophore. So this is motile by cilia. Trichomonas is a flagellate protozoan. But Toxoplasma belongs to the phylum Apicomplexa. It has no particular organ of locomotion, right? So our answer here is A3, B1, C3, B, let me see, which is the answer, A3, right? This is our answer, right? So that's the morphology of Balamuthia. These are motile by pseudopodia. That's the morphology of Balantidium coli. You cannot appreciate it, but if you look closely, its surface is covered by very slender cilia. And that's our Trichomonas vaginalis. Those are the 4 anterior flagellae, and you can see the 5th one folded along the undulating membrane. So these are the 3 phylums of the protozoa. Sarcomastigophora, which contains the subphylum Sarcodina and the subphylum Mastigophora. Sarcodina are the pseudopods, which contain the Entamoeba histolytica, Acanthamoeba, Neglaria, and the Balantidium. These 3, what are they? Free-living amoebae. Then the non-pathogenic ones are Iodamoeba and Endolamax. Moving on to the flagellates, Leishmania, Trypanosomes, Giardia, Trichomonas, Scylomastics, Enteromonas. These are the non-pathogenic ones. These are all motile by flagellae. The second phylum is Celiophora, and the only ciliate infecting man is Balantidium coli. The rest of them are all belonging to the phylum Apicomplexa, the non-motile ones. Plasmodium, Babesia, Toxoplasma, Sarcocystis, Cryptosporidium, Cystoisospora, and Cyclospora. Match the following parasites with their hosts, right? So this is Diphyllobothrium latum. The longest tapeworm infecting man has 2 hosts in its life cycle, Cyclops and freshwater fish. So what do I have here? Cyclops. So A3. Paragonimus, the oriental lung fluke. It has 2 hosts, the 2 intermediate hosts. The first one is snail. The second one is crabs or crayfish. So Paragonimus goes to crayfish. Brugia, the vector is Mansonia. And Fasciola hepatica and gigantica, the sheep liver and the giant liver flukes. These have 2 intermediate hosts. One is snails, the second one is freshwater plants, right? And sheep also can act as definitive host apart from humans, right? So our answer is A3, right? That is our answer. So very important to remember all the hosts in the life cycle. In which of the following situations will you use a sanitizer? So increasingly we are being asked sometimes about contact precautions, droplet precautions. This time we got a question on standard precautions. All of the following, okay, in which of the following? Before examining the patient, after examining the patient, after touching the patient's surroundings and hands are visibly soiled. In this case, you will wash your hands with soap and water. In all the other 3, you can just get away by using an alcohol-based hand rub, right? So your answer is A, B, and C, right? So standard precautions are the minimum infection prevention control practices to be followed in the hospitals. Which are these standard precautions? Performed hand hygiene. Always we will find out how do we perform the hand hygiene. Next is always use personal protective equipment whenever there is a risk of body fluid exposure. So wear your mask, your gowns, your goggles or face shield always. Okay. Use and dispose dispose of sharps safely, perform routine environmental cleaning, clean and reprocess shared equipments in hospitals like your stethoscopes, the BP machines, follow respiratory hygiene and cough etiquette, use aseptic technique, and lastly, handle and dispose of the waste and used linen safely. Right? These are the 8 standard precautions which must be followed at all times for all patients in all situations, because of the belief that all these fluids, blood, other body fluids, leaving out sweat, which regardless of whether they contain visible blood or not, these are infectious to us, contact with non-intact skin or mucous membranes. So with this belief, we are going to follow the standard precautions always in the hospital. Moving on to what are the WHO's 5 moments of hand hygiene, 3 of which were mentioned in our options. These are before touching a patient, after touching a patient, after touching the patient's surroundings. Look at all these. The bed rails, this table of the patient, all these would be patient surrounding. Then after body fluid exposure risk, right? Like you're performing nasal care, oral care, you're, securing the endotracheal or the tracheostomy tubes. And finally, before clean or aseptic procedure. These are the 5 WHO's moment of hand hygiene. So hand hygiene in hospitals, there are 2 techniques. Either use alcohol-based hand rubs or use hand washing. So in all situations, you're going to use alcohol-based hand rubs. Why? Because these can be applied because these are less drying. If you continuously keep washing your hands are going to get dried up very fast, and you're going to have to keep on applying hand lotions very frequently. So in most cases, we are going to use alcohol-based hand rubs, but with following exceptions. Where will you wash your hands? When they are visibly soiled. That was our answer to this question. After using the toilet, before eating or touching food, and finally, when you're handling patients with diarrheal diseases like Clostridium difficile, noroviruses, rotaviruses, in such cases, in these 4 situations, you're going to wash your hands either with medicated or non-medicated soaps. So this brings us to the end of our discussion. Now, before I wind off, a small piece of advice to all of you. So you must be wondering, why are all the faculties advising us? So actually, we are not just your teachers to teach you a particular subject. We are your well-wishers. You are like all our, bachas. We all wish the best for you, that you all do amazingly well. So let me give you a small piece of advice at this time. I know the postponement of the NEET exam has been very disturbing for all of you. You all are very uncertain when is the exam going to be, but at least you have a tentative date that it is going to be somewhere in March or somewhere in April. Most likely it is going to be in March. So March means you've got extra 2 months. How much is 2 months? I always used to study. I used to count the number of days. So it is 60 days. And if you're going to devote at least, I hope you all are going to devote 13 to 14 hours of study at this time. Don't be shocked. This is what is expected of you at this time when you want to fulfill your dream of achieving your dream PG, right? So at least 13, 14 hours, I would suggest you should give. So if you multiply 60 extra days into 13 to 14 hours, it comes to almost 800 hours. This is huge. You've got 800 hours of extra time to prepare for your exam. Like if somebody's, you have to reach the final for say a marathon, you have to reach the finish line in 8 hours. You've been given 2 hours extra to achieve the same target. Isn't that amazing? I feel so ecstatic for all of you that you all got extra time. So for those students who have already prepared very well, had just not managed to make it into the INI-CT exam, for you people, this is an extra time where you can just boost up your preparation. For those people who had not been able to finish your revisions and have got not a very good rank in INI-CT, that means this is the time that you can finish off your revision by December, and then next 2 months just work 14-15 hours and catch up with the people who have done very well, right? And for last group of students who were not able to study very well because of their hard internship, this is the time you've got another 60 days extra, total at least 100 days. Make a very planned schedule for that. Maybe you can just scrape through and get your dream branch, right? So a small advice to all of you. If you're feeling lost, a small advice from my side. This is just my personal opinion, how I would handle this situation. I would say by December, finish off your revisions, and Jan and Feb, just devote to taking 2 grand tests every week. So fix your days, Monday and Thursday. Take 1 grand test each, maybe of Marrow or maybe of any other institute. And on the day, say, do it in the morning itself when you're fresh in your mind and you're all set. You get, used to that exam-like situation. So take your exam on that day, then review your answers on that day itself, all your answers on that day. Find out which are the weak subjects in that grand test and devote the next 2 days on revising either the question bank or the revision videos of those subjects. So you're done. Monday, Tuesday, Wednesday, you're done. Come on to Thursday. Your next GT is due. Take it in the morning, review the questions, and then next 2 days, do your weak subjects. On Sunday, do something that just, you feel this is your total week. You've done your GTs. This is my weak subject. I'm going to devote the whole of Sunday on those 1 or 2 subjects. And you're back to Monday and you're doing your GTs. So in those Jan and February months, you are going to do 16 GTs and you're going to be able to review how many questions, more than 3,500 questions you're going to review. Can you imagine how much you're going to achieve in that much time? It is going to take your revision to the next level. So please, I would suggest you do that. Rest, everyone finds their own way of, cracking their PG entrance exam. Doing GTs is extremely important. How can you go into a war without knowing where you are standing against your opponents? So if you don't know your level, what is your percentile, how can you be, confident that you're going to get your dream branch? So, and of course, you should know how strong the others are in front of you, because after all, you're all competing with each other for that particular rank, right? So knowing your standing is very important, because I read a few of the students' messages in our group in Marrow Links that I had done my complete revision, but still I got such a bad rank. Most likely you did not take your GTs, you did not assess your preparation, and that's the reason when you got a shocker when you took the exam. So assessment of your preparation, of your revision strategy is extremely important, right? So now leave the INI-CT result behind, work on these 3 almost 100 days that you've got, like this is the end of the world. This is the make or break time for you guys. So leave those, let bygones be bygones. That was all history. Rethink your strategy. I have given you one strategy. Maybe other faculty strategies may suit you better. Please just go bang on and achieve your targets. And at the end, this is the biggest opportunity. I would have been so happy had this happened to me. I've got 2 months extra time. So leaving you with this, opportunities are like sunrises. If you wait too long, you're going to miss them. So this is your biggest opportunity to realize your dream in the extra time that you have been gifted by the exam authorities. And last but not the least, I can't resist it. I love to, say these things in fact, I keep repeating it to myself still. खुद ही को कर बुलंद इतना, खुद ही को कर बुलंद इतना कि हर तकदीर से पहले खुदा बंदे से पूछे, बता तेरी रज़ा क्या है? What does this mean? For people who did not understand, let me translate it for you. Make yourself so invincible that your destiny comes and asks you, what do you want? All the best.